If you would like to order services from the University of Arizona Genetics Core, please be sure you're logged into the iLab operations software. Once you're logged in and at our core facility landing page, simply click the tab that says Request Services. This will open a list of available services. If you do not see the service you're looking for here, please click Consultation Request, and this will open up a form where you can tell us about your project. To request fragment analysis, click here to expand this category, and on the right, click Request Service. You will first be prompted to select the lab that you're a member of. This will open up the form for a fragment analysis service request. Please be sure you complete all required fields indicated with the red star. This plate name is a user-defined field and may simply correspond to how you have your plate labeled. This dilution factor will apply to the entire plate. We'll dilute your samples once they get here, so please simply select the dilution factor you'd like us to use. If you're uncertain, select 1 to 50, as this is the default and is suitable for most situations. Next, you'll be asked to select the dye set. We offer 4-dye and 5-dye fragment analysis. If you're uncertain which is best for you, please contact our core facility, as this may affect your results. Once you select the dye set, you'll be prompted to select which size standard or ladder you'd like to use. GeneScan 500 is the standard ladder and applies to most situations. Please select this one unless you are certain you want an alternate ladder. Next, you're going to be asked to provide us your sample names and the well position that they may be found in the 96 well plate. You can enter these one at a time, with the corresponding well position here, but you may find it easier to paste these in. You have several options. One is you can download this simple list to Excel, fill it in and upload directly to the list. Or if your samples are arranged in a graphical plate map, click our converter here to download. When you download and open up this converter in Excel, please follow these simple directions. First, you will paste your samples into this plate map here. I'm going to open my PCR sheet also in Excel. Here's my PCR sheet, and this is the plate map that I use to plate my samples for PCR. I'm going to copy all the sample wells, but not the coordinates on the outside of the wells. Go to the plate map converter and paste into the fields here. You'll see that my sample IDs and their graphical plate map orientation have now been translated into a list with corresponding well locations. You're going to copy this list, none of the headers, and paste this into your iLab's form. Please be sure to copy all records so that our lab can have them as well. Select both columns and simply paste. This will paste in all 96 rows that we copied out of our converter table. Take time to ensure that your sample IDs are positioned in the right well, because this can affect your results. Scrolling through, I'm verifying that my sample IDs are listed with the correct well location. When you're satisfied, let's go down and finish filling this form out. Next, you're going to be asked to enter the total number of samples. For this service, we bill per sample, so if you have wells that are indicated as empty, they will not be billed. My submission has 2 empty wells down here at the bottom, and I'm only submitting 94 samples. Once you enter the number of samples, click Add Selected Services. This will calculate an estimated project cost for the quantity of samples and the price per sample. Please take the time to acknowledge this statement here. This explains that the UAGC will keep your project materials and supplies for 90 days following project completion. After this time, we'll dispose of your samples in keeping with our SOPs. You'll be asked to agree to this each time you submit. This field here shows the total projected cost. Unless there's additional services requested, your project cost should be as indicated. Finally, you'll be asked to indicate how you're going to pay for this. If you have a U of A account number, you can select it from the drop-down list here. If you do not have a U of A account number or you're an external user, you'll be prompted to enter a PO number or indicate whether you're paying with a check or credit card. If you'd like to split this charge between multiple accounts and you have access to multiple accounts, click here. This will indicate how you're able to split it, and keep in mind your percentages must add up to 100% in order to order this service. This should complete all necessary form fields. Feel free to take the time to review, and when you're satisfied, click Submit Request to Core. When you're back on your dashboard, you can see the submission that you just created. This is a fragment analysis submission. It is very important that you copy down the numbers that follow the letters in your service ID and transfer this to your physical submission. This will be how we identify your physical submission and match it to the electronic submission when it arrives in the lab. If you have questions about how to use iLab software to create fragment analysis service requests or to request other services from our laboratory, please email us at uagc@email.arizona.edu. We look forward to working with you.